What I want to tell you about is a study that really started about 15 years ago through wild bird surveillance in Kazakhstan. And it was started by my colleague who I met actually last week. He was attending a parasitology conference at the University of Kent, and we managed to meet up and make plans going forward. We've got Sept here, also his collaborator at the Frederick Loeffler Institute in Germany, Tim Parder, and of course the people that are going around sampling all these, all of which I can't away names. But I just want to acknowledge all the work that we did and my small contribution, which you'll see. Good try. Let's click on the screen. Very good. Action. Yeah, I just tried to get. Yeah, try that. Anyway, right. So, brief overview. As you've heard, there are 4 main types of influenza: A, B, C, and D, and they're illustrated by the colours circles that you see on here. You can see there's a vast number of different both avian and mammalian species that are infected by these viruses. But it's only humans and pigs that have shown susceptibility to infection by all these 4 types, albeit not producing necessarily particularly impactful disease. A and B are the ones that we associate with seasonal flu in the 2 hemispheres in the winter months. And currently there's a quadrivalent vaccine, 2 circulating A and 2 circulating B strains. It sounds like they might be dropping the other B one though. So we may go back to a trivalent vaccine. And of course, as we heard earlier on, it's the type A that produces these severe sporadic pandemics throughout the world. That's illustrated succinctly this morning by Spanish flu by John Oxford here. And of course, within living memory for the rest of us in 2009, the swine flu epidemic, which raged around the world, luckily didn't have the kind of death toll that we saw with the so-called Spanish flu. I can really zoom through this, but these are basically the mechanisms behind the rapid evolution of influenza viruses are twofold. Firstly, drift. These are RNA viruses that have poor proofreading ability of their RNA polymerase, and they're changing all the time, very much like we saw for the SARS-CoV-2. It changes the surface antigens, which changes the immunogenicity and the responses to those antigens on the surface, namely H and N mentioned earlier on. There's also shift, where you have the 8 segments of the genome in superinfections recombining to producing new types. And this is one of the main mechanisms for the pandemics that we've seen throughout the past 100 years, but not as mentioned earlier on for the Spanish flu. So different mechanisms going on producing different outcomes. And that's illustrated by this little cartoon here. So, as we've covered in some detail, wild birds are the primary reservoir for these through migration, congregation in large bodies of water, and the categorization of these viruses are due to these surface antigens, haemagglutinin and urominase. And you've heard about the role that they play. So there are 16 HAs, 9 NAs, and global monitoring of the circulation of these viruses continues, and we've had a very important rationale for why that needs to continue. And we're going to touch on that kind of surveillance just in a minute. And of course, we've also mentioned the fact that 2 novel influenza viruses were found in Central and Southern America, within the last decade, namely H80 and H70. So we have to be aware that there's a new evolutionary route appearing here. So migration of these birds throughout the world carrying these new subtypes and new combinations of H and N subtypes is a crucial way for this to travel and be spread and potentially move into animals and also into human beings. So this map, just showing coloured arrows, is really giving you some of the main migration routes. But of course, when we break these down, there are other migration routes also. You'll see them. So, complex transmission patterns, wild bird reservoir, passing over into other species, which can be due to mutation, shift, and drift. There are human-adapted H1, 2, and 3 subtypes which circulate, and indeed in pigs as well. And some avian influenza subtypes, as you've heard, have infected humans, namely 5 to 7, 9, and 10. The last avian subtype that was identified was in 2005 by Fouchier's group, who was mentioned earlier on, and this is H16. And the complexity of what's going on out there in terms of ecology is illustrated with this diagram here, which is being added to all the time. So we've got a complex, constantly evolving system. Now on to the particular study that I got involved in. Well, prior to me getting involved in this, there was a huge wild bird monitoring study carried out in Kazakhstan between 2002 and 2009, which produced almost 5,000 biological samples and represented about 150 different wild bird species, predominantly those that are waterfowl and shorebirds, because this water can actually return wrong. Virus isolation from influenza positive tested samples was carried out using embryonated chicken eggs and then passed on into cell culture, which is a routine system. And these isolates were then tested in HA and neuraminidase inhibition assays and published in this paper. So back to those migration routes, a little bit more detail that we see here. In the center, you can see Kazakhstan and a large body of water, which is on the left-hand side of the right-hand image. That's the Caspian Sea. And on the diagram underneath, you can see where the sampling was carried out. It's particularly this area with the red flags on the right that's I want you to concentrate. Because of the 95 positive samples of those 4,800 biological samples, 58 of those came from the Caspian Sea area. These subtypes were confirmed by RT-PCR against the nuclear protein gene and then sequenced. But interestingly, a few of these samples could not be isolated, even though most were. Those that were revealed 8 subtypes, predominantly H30, but also 3, 4, and low and H5. And there was some evidence of species and geographical patterns occurring within the samples that were taken. But one particular sample was of specific interest. So this was an RT-qPCR positive cloacal sample. It was named KZ52. It was given the ID KZ52, and this is from the common pot chart taken at the end of 2008. Initial sequencing of PCR products from this couldn't classify it within the existing subtypes. So whole gene and genome sequencing was attempted to see what they actually had in there. Unfortunately, at the same time, virus isolation from these samples was unsuccessful despite a number of different attempts. And what, talking to Sept last week, he was saying is that the transmission of these samples from the wild in Kazakhstan from one lab to the other, and then the taking of freezing, taking them out of the freezer to do various testing. There's a number of freeze-thaw cycles, and we think we've got evidence, because we can't get anything more out of these samples, is that that's caused deterioration. And that may be something to do with virus isolation was difficult as well. So RNA was extracted from this sample. Reverse transcription PCR using universal influenza primers was conducted, based on the Hoffman paper a couple of decades ago, to amplify those genomic segments. The sequencing of these individual genes on these segments was carried out, and it was only the haemagglutinin that produced a whole length open reading frame. There were partial sequences of NP, NS, and PB1. And interestingly, no PCR products representing NA were fished out of this sample. So that may or may not indicate that there's some divergence with the other index here. We're yet to find out. So that was conducted in the University of Vienna Medical Veterinary School by my colleague Sept. And in parallel, we did a different approach to the sequencing, which was in effect HA specific. We concentrated on that. We used gene walking and nested PCR to walk along the open reading frame till we had the entire sequence, which was very, very good. So the auth was 1,686 nucleotides in length. And then using those sequences, it confirmed what was found in the other lab, exactly each nucleotide. So then phylogenetic analysis was carried out both in Vienna and in Germany, by comparison to 91 representatives of the 16 avian and 2 bat influenza H subtypes using MEGA11 software. And this was in fact published earlier on this year, as you'll see. So just to boil down the results, this is what was found. So KZ52 is sitting in the middle here, and its nearest relative was H9. Slightly further down the tree were H8 and 12. So this means that KZ52, this HA subtype, this HA type, fits into the group 1 influenza viruses shown in blue in this little diagram. Simplifying that diagram, we see KZ52 in the middle and quite clearly the HA9 branching on the other side of that particular tree. And talking to my colleagues, what this represents is, if you add the length of the trees, of about 600 nucleotide changes within that about 1,700 nucleotide length of the open reading thread. So if you then take the amino acid sequence of this HA, 561 amino acids, and compare it to its nearest relative, so a number of different H9 types, this is what we see. I know you can't see it very clearly, but how they align together. And overall, this has told us that there's about 68% nucleotide and amino acid sequence identity between KZ52 and H9. So it's quite diverged from the original H9. The cleavage site, which was mentioned in an earlier talk, where you have the cleavage of the HA, is positioned where that little asterisk is in the middle section of the diagram. And this will become relevant in a minute when I talk about pseudotyping. And finally, if you look at the identity of the other partial sequences we've got, they're much higher, they're in the 90s. So the HA is very different than what we're seeing for the other genes, albeit very partial sequences. You can have a look at that detail within the paper itself. So this is a 3D model we constructed based on KZ52 with the green and yellow HA1 and H2 parts of the HA, the cleavage site shown in red, and the receptor binding site top in blue, which binds to the sialic acid regimen. So the question is this a new subtype? Well, subtyping isn't based purely on genetic analysis, it's based on antigenic properties. And this is done typically using immunodiffusion assays as published by the WHO in the bottom right-hand corner. And an example of that is from the Fouchier H16 paper you see here. And you can see the serum, paper-immune serum in the S slot, and you can see the antigens in the A slot. And you can see with that little red arrow that there's this precipitated antibody antigen showing that line there. So of course this needs to be done, and it will need to be done for NA as well once that is found. Okay, so we didn't have the isolate of the virus, so the question is, what can we do to continue the research in the absence of that isolate until that comes out? And the obvious thing to do was to make a mimic, a surrogate, and that's where pseudotyping came in. This is, as you've heard earlier on, has been very useful for all sorts of different virus families. And of course, it has the core of one virus, the native or the active virus, and the envelope of the other, which of course would include the KZ52HA. And reasons, apart from the fact that you haven't got the virus, you can work with these in low containment, they're easy to do mutation studies, etc., etc. So that is the wild type virus cartoon. And this is the pseudotype with just the HA shown on the surface here. Internally, we've got the lentivirus components here. This harks back to Kelly's talk a minute ago, showing you the group 1 influenza viruses that we have in the portfolio within the lab, showing H9, its relative here, and indeed H18, that virus solution as well. So transfection experiments, as Nigel mentioned earlier on, taking the components of the pseudotype as plasmids, transfecting to produce a cell line. So you've got your core, you've got your reporter, you've got your HA, and you've got your necessary protease. But of course, you don't know what the protease is at this stage, so we have to time them. And then you cause release of the particles once they assemble, adding neuraminidase, typically from Clostridium, although you can make pseudotypes with the NA combined actually in the particles themselves. Then you harvest the virus, you can put them on a susceptible target cell line. Again, what is that susceptible target cell line? Is it what we've seen in other influenza viruses? Is it something different? And of course, you can do neutralization assays by mixing serum with the pseudotype particles and look for a reduction in reporter gene expression on target cells. So, where did we start? We started with the native HA or reading frame. We also created a codon optimized version of this because the producer cell lines is a human cell line, trying to increase the expression of the HA. The cleavage site, when you look at it, is actually identical to H18, which of course we have as part of our portfolio. Its closest relative is H9, and both 9 and 18 have been pseudotyped using 10'4 protease for that cleavage event. So of course that was our first port of call. But subsequently we also tested other proteases, 2, 3, 4, 6, HAT, kallikrein 5, and basic trypsin, all to try and cause this cleavage, which will allow, if you see on that tiny diagram, the escape of the virus particle from the endosome to allow it in nature to replicate. And we use H9 and indeed H3 as controls. But this is the cleavage event that occurs, producing the fusion protein which is necessary to escape from the cell. So here are the results. So as you can see, firstly from a crude look down a microscope, at the GFP being used as the reporter gene, only the H9 and not the KZ52 showed transduction with the particles on 293 HEK cell lines. H9 and H3 work, so this is a luciferase readout in the graphs here. So that was a disappointment. The question is, are we choosing the right target cell line? So we've got other target cell lines that we use for many different influenza virus experiments within the lab, NDCK1 and 2, a duct embryo fibroblast cell line, and indeed also HEKs expressing constitutively proteases. And with those, we saw no transduction for KZ52, unlike the H9 and H3 controls. So is there something weird about this cleavage site? So the consensus for H9, if you look across thousands of different H9 strains, is this PARS SSR GLF. So what we decided to do is to mutate the KZ52 cleavage site and move it towards H9 and eventually get to H9. Conversely, we were going to do the same thing back for the H9 pseudotypes into KZ52. And also, belt and braces, we changed the cleavage site to a multi-basic site where ubiquitous proteases in many cell types will cleave it, just to see if we can get something out of this. And what we basically saw is that we couldn't make functional pseudotype particles using this that would transduce the cell lines. However, if you put the KZ52 cleavage site into H9, it did work, and it did, so they were functional particles. The H9 also worked with the multi-basic site, although in our test, it was at a lower titer. Again, it didn't work for KZ52. So unfortunately, this is a story of woe, but there is a little glimmer of hope at the end of it. We tried a different core because we have found different cores with different impacts in terms of the type of pseudotypes you get out of it. So we turned to VSV, the rhabdovirus core, and again, H9 worked with KZ52D. So the question next was, are we actually getting expression of this HA on the surface of producer cells? Because unless the core can go out through and bud out through the producer cells and get wrapped in the envelope, with that HA on it, you're not going to get functional particles. So is it there? So we don't have KZ52 antiserum. So what we do have is from NIBS is H9, described as an H9 light sheep polyclonal antibody. So we use this with the hope that we'd have enough cross-reactivity to we'd actually see something. And lo and behold, our H9 control worked, our KZ52 at a lower level, we did detect something, and indeed one of the other closer relatives, H9, also showed some signal on a par with what we saw for KZ52. So it looks, and compared to all the controls, so it looks like there is at least some of that HA getting to the surface of the producer cell lines. Is the lower level due to the fact that it's cross-reactive and isn't as punchy as H9 matched with H9, or is it at the lower level? And that's the problem we've got. And actually, if you look at it, it's a higher magnification. You can see the halos on the membrane on the outside of the cells. We then turn to ELISA. And what we did here is we got the particles, we purified the particles, in this case, with a Lenti-X concentration system from Takara. We then did a protein assay so that we got antigen loading correct in the ELISA. And then we used the H9-like NIBS anti-spirit again as the detection system. And what this shows, albeit at a lower level, if you see the red line compared to H9, H9 is nice and high here. The delta envelope, no surface glycoprotein in grey, but for KZ52 in red, we are seeing something. So it looks like the HA is on the surface of pseudotype particles. So in a binding assay, we're seeing something, that chink of light in the darkness, but it's not functional biologically, or at least we haven't got our system working so that we can detect it. So I'm not giving up, but you know, this is, people seem to think pseudotypes are click and play. You produce them, you get them, you do what you want with them. And we've known through history, through producing that portfolio, that there are some really hard nuts to crack in there, and sometimes you don't get them out. But I think we've got enough light here to show that we're moving in. So, I've got enough time just to go through the summary. Yeah, okay. So, novel influenza gene sequences were detected in this particular pot chart in bird surveillance in the last decade. The complete HA was sequenced, and along with partial sequences of 3 other genes. The identity for the KZ52 HA compared to its nearest relative H9 was 68%. No virus has been isolated to date, so we need to continue to look for that. Attempts to generate pseudotype particles were myriad, different approaches, not all of which I've mentioned here. Functional H9 pseudotypes were produced, but not functional, biologically functional KZ52 pseudotypes. But we did see that protein on the surface of the producer cell, we think, and it appears to also be on the particle envelope. So we're now pursuing further approaches to try and open up this window and get something out of it. Because until we actually have the virus from nature, this is the tool that we've got. So a little bit of background in italics here, we've covered this already. But what I wanted to point out to you is that the H9, as we heard earlier on, is endemic in many countries and has been isolated from man, mainly children. The WHO now consider H9N2 to be a pandemic concern. And of course, our closest relative is H9. H17 and 18 have been detected in Central and South America, and I think Nigel just hinted earlier on about the study in Nigerian bats, where we've actually got some hits I was telling you, yes. But there's no interspecies transmission noted today. But of course, we know that lots of zoonotic viruses come from a bat reservoir. H9, however, has been detected in Egyptian fruit bats, and these bats could be experimentally infected as well. How H9 relative KZ52 fits into this changing picture, we don't know, but we need the detection systems, the experimental tools in order to go out there and find out where this virus is. Until we've got this virus, we can't really evaluate in any great extent how it fits into the ecology. But we will get there, and we are starting to produce tools that enable us to go out and try and fish it out back in nature. So fingers crossed. Okay, thank you very much for your time. I just want to leave you with some bits of the team that involved in that particular study.